second-year Master's of Environmental Science student on the Student Planning Committee. BIOMES is YSE's flagship seminar series, bringing you cross-cutting topics in environmental science. Today, I'm delighted to introduce Dr. Michelle Wong. Dr. Wong joined Yale as assistant professor in the Department of Ecology and Evolutionary Biology this past year. Dr. Wong is an ecosystem ecologist and biogeochemist. She studies how plants and microbes interact with nutrient availability to shape terrestrial ecosystem response to global change. Her research asks how terrestrial ecosystems overcome nutrient limitation to facilitate carbon storage in plant biomass and soils. Dr. Wong was formerly a postdoc at the Carey Institute at Ecosystem Studies. She received her PhD in ecology and evolutionary biology at Cornell and her bachelor's degrees in molecular environmental biology and sociology at UC Berkeley. Please welcome Dr. Michelle Wong.
Thanks. Thanks, Phoebe, for the introduction and for the invitation to come speak in the BIOMES seminar. And I apologize, I haven't been able to attend the seminars this semester because my class is at the same time. But today I actually brought my ecosystem ecology class, so wave hi to them, they're in the back. Yeah, and it's great to be here, and hopefully there's a lot of overlap, I think, with some of the work being at EB and also in YSE. So it'd be fun to chat more with students and postdocs who are thinking about nutrient cycling or forests or root strategies. So probably most of you know this, but forests play a really critical role in mitigating climate change. And currently we're emitting, for example, about 11 gigatons of carbon into the atmosphere every year, which is equivalent to about 5 parts per million of CO2 in the atmosphere every year. And for context, about 2 gigatons of carbon is equivalent to about 1 ppm of CO2. But only about 40% of this carbon, give or take, actually stays in the atmosphere, causing climate or exacerbating climate change, because about a third of the carbon that we emit is actually taken up by the land carbon sink, and the rest by oceans. And this is really due to increased rates or even increased efficiency of photosynthesis. And temperate and tropical forests actually play a really big role in this land carbon sink, especially regrowing and mature tropical forests, although this land sink has been shifting more towards boreal and northern temperate forests. Well, what about into the future? So the largest projected carbon sink is in the tropics, at least based off of modeling projections. And this is a modeling projection in response to a change of 250 parts per million of CO2. But there's also quite a bit of uncertainty across the tropics as well because of this large projection. And so one of the big questions that a lot of us ask are if the land carbon sink is going to continue to take up carbon at the same rates indefinitely, or if eventually they'll be limited by resources. And two of the most often constraining nutrients or resources for carbon uptake are nitrogen and phosphorus. So nitrogen is really important in nucleic acids and amino acids, especially in proteins such as rubisco, which catalyzes photosynthesis. And phosphorus is also required in nucleic acids and in energy compounds such as ATP. And really, nutrients can limit productivity because the soil doesn't have an endless supply. That's why we use fertilizers to grow our food, and that's why the production of fertilizers have really allowed human populations to double in size. And when these larger Earth system models project the land carbon sink into the future and they incorporate nutrient cycling, they find a decrease in net primary production of about 19 to 25%. So here the different colors, red and blue, indicate model projections when they incorporate nutrient cycling, and the black line indicates modeling projections when they don't include nutrient cycling. So this kind of flips the land carbon sink from a sink to a source also in about 100 years, give or take. And nutrient limitation, not just in terrestrial systems, but also in freshwater and marine systems, is quite prolific. So this is a meta-analysis showing response ratios of ecosystems that are different pot studies that have shown responses to either nitrogen or phosphorus additions in relative to just control treatment. So you can really see that nutrient limitation is widespread. And now we have tools to be able to make some of these gridded data sets. And this is a projected nutrient limitation response or projected nutrient limitation of both nitrogen and phosphorus across terrestrial ecosystems. And you can really see that nitrogen is really thought to be extremely limiting in northern temperate regions and then phosphorus across the tropical regions. Although maps like this also don't really account for a lot more heterogeneity of other nutrient limitation, as well as temporal variation in time, which is what we'll talk about today. So a lot of the avenues that I ask questions in are kind of related to 3 different questions, all to get at understanding terrestrial nutrient limitation. So the first asks, how are nutrients distributed in the environment? The second is, how does nutrient availability change after disturbance events? And the third is, how do plants and plant communities address and actually overcome nutrient limitations? So to what extent can they respond to and modify their environment? And a lot of the work that I'll talk about today have involved a huge slew of collaborators who I won't be able to name all of them today, but they are listed here. And I'll also talk about work in 4 different regions indicated by the yellow triangles. So the first topic is kind of just really asking how nutrients are distributed in the environment. So most of the biological or potentially available pool of nitrogen available or eventually available to plants and to organisms is actually in the atmosphere. So about 78% of our atmosphere is composed of dinitrogen, which is super, super stable in the atmosphere. And the main mechanism of how this nitrogen comes into the environment is through nitrogen fixation. And there's predominantly 2 pathways, which we group as symbiotic N fixation, which occurs through a symbiosis between microbes and specific plant species like legumes. Soybeans are a really great example of a nitrogen fixing plant. And the other pathway is through free living nitrogen fixation. So this is mostly catalyzed by bacteria and archaea, usually heterotrophically. And 80%, at least from modeling projections of this global nitrogen fixation, is thought to occur in the tropics. So really tropical forests are thought to be a hotspot of nitrogen fixation. And really for 2 reasons. One is that there's a high abundance of these leguminous nitrogen fixing trees in the tropics, or at least in the neotropics in the Americas. And the other is just there's a lot of productivity and a lot of decomposition that can facilitate high rates of free living and fixation in the tropics. But nitrogen fixation as a process also has nutrient constraints on its own. And 2 of the most common limiting elements or thought to be limiting elements for nitrogen fixation are molybdenum and phosphorus. So phosphorus is required for ATP, and there also could be a bias in the sense that when we add phosphorus to test for nutrient limitation, we're also shifting ecosystems away from nitrogen limitation and towards phosphorus limitation. So that could be one reason why phosphorus tends to stimulate nitrogen fixation. And the other is that molybdenum is a cofactor in the nitrogenase enzyme. And historically, phosphorus was thought to be the strongest constraint on nitrogen fixation. So plenty of studies have shown nitrogen fixation responses with phosphorus additions, but a recent or relatively recent study showed that there's actually a lot of molybdenum contamination and a lot of traditional phosphorus fertilizers. And so it's a little bit hard to disentangle historical studies that have added like triple superphosphate because of that molybdenum effect. And new molybdenum and phosphorus and pretty much every other rock-derived nutrient comes into the environment through rock weathering of bedrock, eventually into saprolite and then into soils. But molybdenum is really interesting because it's actually rare in bedrock. So its crustal abundance is about 1 part per million. And also when it's oxidized, it's very, very soluble. So it tends to leach away from soils or oxic soils relatively easily, and then drains into streams and then into rivers and into oceans where it has one of the highest concentrations of any trace metal. So molybdenum has this really interesting distribution where it's kind of low relative in soils and then more abundant in freshwater or in ocean pelagic systems as well. And so basically, we're set up with this system where in temperate soils, we might have less weathered soils due to a lot more recent glaciation events, and then also less physical and chemical weathering that we get from higher temperatures and higher precipitation in the tropics. So there's this interesting dichotomy where theoretically, there should be relatively low molybdenum and low phosphorus in tropical forests, but we theoretically also have high nitrogen fixation rates. So where do tropical forests then get this molybdenum and phosphorus to support these theoretically high nitrogen fixation rates? About 80%, again, is thought to occur in the tropics. Okay, so where do tropical forests get molybdenum to support N fixation rates? And we have this hypothesis that atmospheric sources could be a source of molybdenum. So like I mentioned earlier, sea salts are high in abundance in molybdenum concentrations. Volcanoes also emit aerosols. And then also mineral dust is one of the most important sources of atmospheric nutrients or movement. And this may be news to you, but every year wind blows from the Sahara annually, which gets entrained into the atmosphere. And then although it may seem like unsubstantial amounts of nutrients, it's been linked to contributing iron towards the Atlantic Ocean for phytoplankton, and then also for phosphorus to the Amazon. So about the same amount of phosphorus that gets deposited via Saharan dust to the Amazon every year is also what gets leached out through the Amazon River as well. So there seems to be some steady state impact occurring with this Saharan dust. And so this may be news to some of you, but most people did not care to measure molybdenum in the Saharan dust. And so we actually had to go and work with some collaborators where we collected samples from across the Sahara, specifically concentrated in the Bow Valley Depression, which was historically thought to be a hotspot of the Saharan dust. And we measured molybdenum concentrations in the dust. And then we also parameterized this model. Molybdenum was really well mixed in sea salt sprays. And then we also used a molybdenum to sulfur ratio to estimate volcanic emissions. And then I'll show you some modeling outputs. So what we found, at least for these natural sources, was that dust comprises about 70% of natural molybdenum deposition. The warmer colors here indicate higher deposition rates, and then the black triangles and circles indicate some of the dust and sediment samples that we collected across the Sahara. And the scale is not linear. And then when we add in sea salt sprays and volcanoes, this is the modeling output. And really the main takeaway here is that molybdenum deposition is not even across Earth's surface. And the other thing I wanted to point out is that the units are in milligrams of molybdenum per meter squared, which is very, very small. That's probably less than just a sprinkle of salt like on your toast in the morning. But the rates between 0.01 and 0.05 were actually the fertilization rates that a previous study in Panama used to stimulate nitrogen fixation. So even really, really low amounts of molybdenum can have an impact on relieving any molybdenum limitation for nitrogen fixation. And the other thing that we noticed was that when we actually layer historical nitrogen fixation studies or measurements across the tropics, these are indicated by this black circle or the black circles here. We found that, and probably no surprise to anyone in this room, the studies were also not evenly distributed. And here I wanted to orient you towards the Amazon basin, which, you know, from those modeling projections is thought to be this hotspot of N fixation, indicated by this blue circle here. And you can see that the northern Amazon basin receives much higher molybdenum deposition than the lower Amazon basin, indicated by that yellow to blue color. And so we wanted to measure molybdenum and nitrogen fixation in this region, indicated here by this yellow triangle. And so our first simple question was, is molybdenum low in the southeastern Amazon? Theoretically, there's high weathering rates across the lowland tropics, or that should have depleted some of this rock-derived nutrient, and also there's low deposition rates. And this is Santorum pulling out a soil auger going to 10 meters, and then this is me in a soil pit as well. So you can really see how weathered these soils are. There's very little rock input. And I don't know if anyone's cored soils in the tropics, but this is it's at least at 10 meters now. So very, very weathered soils. Okay, so I basically plotted some of these other tropical regions where people have measured molybdenum concentrations, with the purple indicating the average crustal abundance. And to no surprise, the molybdenum concentrations were relatively low. And the same for phosphorus. So like we expected, low molybdenum and low phosphorus. The second question is then we want to understand if the nitrogen fixation is actually constrained by molybdenum. So to do this, for my dissertation, we set up this 60 plot fertilization experiment, 2 by 2 meters. And this is Giovanna, who's a student with Mercedes Bustamante with a limed plot. So we also added molybdenum and phosphorus in combination with lime, because we also know that the soils are really, really acidic across a lot of the tropics. And the acidity can also further bind a lot of anions, making them less available to microbes and to plants. And these are just 2 examples of time points. So yeah, so we fertilized, and then we expected that nitrogen fixation would increase with extra amendments. And then we measured free living and fixation with the acetylene reduction assay in both litter and soils. And so this is just a response in the wet season after 1 week of nutrient additions, and then after a month. And we did see a slight response to molybdenum, phosphorus, and lime additions in the wet season after a month in soils, but not in litter. But in reality, actually, we checked and we measured fertilization across many, many time points. And this is a lot, obviously very complicated. So I'll just show a simplified version of this. So here, the green just shows any significant response and to any of these amendments across different time periods. And so you can really see that we only saw responses very, very few times, just 2 times in the soil. And this was really, really surprising because pretty much every other tropical study that had added molybdenum or phosphorus separately had found responses to either amendment or to either nutrient addition. So in Belize and in Costa Rica, they basically typically always found some response. So why not in the southeastern Amazon? Why not at this site? And we hypothesized that it might be just due to extremely high nitrogen availability that is uncharacteristic of some of these other regions of the tropics. And so here I have a plot of a C to N ratio here, and basically a high C to N ratio means there's more carbon atoms relative to nitrogen. So that means less nitrogen availability, whereas a lower C to N ratio indicates a higher nitrogen availability, so more nitrogen relative to the amount of carbon that's available. And you might expect that it's energetically expensive to fix nitrogen, right? Because it's triple bonded N2. That under lower nitrogen availability, you might expect higher rates of free living N fixation. And when we plotted the stoichiometry and the rate data across a lot of different regions of the tropics, we found that our site in the southeastern Amazon did fall on the higher nitrogen availability end. So some of these other tropical studies did seem to map out in this relationship as well. And the other was, and this is just temporal data with inorganic nitrogen additions, inorganic N extractions. So just temporal data, but just even in the surface soils from 0 to 10 centimeters, we did find higher nitrate and ammonium concentrations as well, sort of indicating, or to some extent indicating nitrogen richness at the site. And I'll go into this in a little bit more detail in the next part of the talk. But in summary, we did find that molybdenum deposition does vary across Earth's surface, and that it probably is really critical to maintaining molybdenum availability in different ecosystems. And like we expected, molybdenum in the southeastern Amazon was low, but that nitrogen fixation was not molybdenum limited. So I think in the future, it's going to be interesting to continue to start unraveling some of the drivers of variation across the tropics, because I think we're not at this point where we think that there's one specific nutrient limiting one specific ecosystem. So I talked a little bit about where nutrients are limiting, but I want to talk about when next, because nutrient limitation can also change over time, especially after disturbance events. And in the tropics, where we have over half of tropical forests in some stage of recovery after a disturbance event, this is particularly critical. And so the question here is, how does fire affect above ground nitrogen? And this is really a critical question because fires are increasing in severity, and nitrogen availability is also really locally controlled. And nitrogen is also preferentially lost in fire events because fire impacts nutrients differently for different elements, especially a lot of nutrients that are concentrated in wood, such as calcium, tends to be redeposited. And it's also on the soils as ash, and also because nitrogen is volatilized at a lower temperature than other nutrients, and because nitrogen is concentrated in leaf biomass. So after fire events, even though tropical forests are not as a whole thought to be nitrogen limited, they can be after some of these events that are disproportionately losing nitrogen faster than some of these other elements. From a previous study in the Amazon, Kaufman et al. Found that about 50 to 60% of the above ground nitrogen was lost from an ecosystem, but only about 7 to 32% of phosphorus was lost. And I'm going to zoom in to this map of pastures and croplands in the Amazon. And I think a lot of you in Paulo Brando's studies actually visited this region. Can you raise your hand if you actually went to Brazil this? Okay, a few of you in this room, awesome, great. So this may be no surprise to some of you, but you can see really the blue and the red colors here, which represent pasture and croplands. And this map is from 2017. So some of these land use cover types may have changed. And basically, this land clearing has huge consequences on the local water cycle because as trees are cut down, they're no longer taking up water. There's less evapotranspiration, and this makes forests a lot drier and more susceptible to drought. And this, in combination with ignition sources from agricultural fires, can cause fires to spread in areas that they haven't been historically before. And I think like Amazon fires were really on the news in 2020. I think we all remember that, but in reality, the fires are getting worse and it's not really on the news now. Obviously, we have a lot of things going on in the world, but basically, this is really, really impacting a lot of different ecosystems that haven't experienced fire before. And I'm going to take you to Tanguru Ranch, which I think some of you did get to visit during that field trip. And some of my collaborators, including Paolo, had conducted this large-scale fire experiment to basically look at interactions of fire and edge effects on ecosystem dynamics. And these are large-scale and experimental burns across 50-hectare plots. And they're really low, relatively low-intensity, slow-moving surface fires across the litter layer. And I was actually trying to find a picture of Paolo, but this was the picture. The first time I met him actually was in 2016 at Tanguru, when I was doing my PhD. And one of the advantages from some of these experiments is that we can track dynamics before and after the fire events. And so we have information about basically the tree inventories and the biomass. So we're able to extrapolate nitrogen budgets from these studies. And what we found was that about, at least using the 10 most dominant species, which comprise, I think, 50% of the abundance of trees, we found that about half of the wood nitrogen was lost after these fire events. And I forgot to explain that there's basically I'm only going to show 2 treatments today for simplicity, but there's a control forest, which obviously hasn't been burned, and then a forest that was burned 3 times every 3 years from 2004 to 2007 and then to 2010. And so the brown line indicates that treatment, and then the green indicates the control. And we also were able to track leaf nitrogen. So we measured the concentrations of leaves in the 10 most dominant species again. And we actually found this increase. So 2010 was the last fire year. We're not really seeing this increase or recovery in the wood nitrogen pool, at least by 2016. But the leaf nitrogen pool, we're starting to see an increase, or we started to around 2011, 2012. And this increase is equivalent to about 5 kilograms of nitrogen per hectare per year. And this number will make sense later. But because we were able to track dynamics before and after the fire, we were also able to see the species that were present. And what we were really surprised to see was that actually N-fixing trees increase in their abundance after the fire events, which is not so surprising because a lot of these N-fixing trees are early successional species, and they tend to do well in high light conditions, and of course after disturbance events to basically fix nitrogen under these theoretically nitrogen limiting conditions. And so we actually wanted to measure nitrogen fixation at this time point where we saw this increase in the abundance of these trees. And so asking, does nitrogen fixation increase post-fire to replace some of those nitrogen losses? And to do this, we measured this time both free living and symbiotic N fixation. And symbiotic N fixation, we actually did 2 approaches. So we used basically a systematic approach because trees can spread their roots tens of meters away, where we sampled in a gridded like per meter grid. And then we also sampled around the base of about 42 trees. And I don't know if you recognize, but this is Leandro, who's in the audience, who's an associate research scientist here. So he actually helped me pick out some nodules at one point in time, which was really fun. So yeah, we did a grid sampling. We sampled around the base of trees, and then we also used the acetylene reduction assay, and then we paired that with an isotopic label as well. Okay, so what did we find? Well, we did find slightly increased symbiotic nitrogen fixation rates in the burn forests, but very, very low rates, so less than 1 kilogram of nitrogen per hectare per year, and essentially no fixation or no symbiotic fixation in the control forest, which was really surprising. Because this number is about 10 times, even in the burn plots, about 10 times lower than a pantropical estimate of nitrogen fixation. And when we add in free living N fixation, again, those rates are relatively low. There's actually higher rates of free living N fixation in the control plots, probably due to just higher amounts of litter fall and then also more moist conditions. So basically, we didn't really find that N fixation was replacing some of those N losses, or that they were meeting the demands in the leaf pool that we saw this increase of about 5 kilograms of nitrogen per hectare per year. And when we compare these findings to some of these other studies that have looked at N fixation across succession, so each dot here is going to represent a different successional age, we found really, really, really low rates of N fixation compared to many of these other forests, which is really surprising considering that we see this huge increase in abundance of nitrogen fixing trees. So why is this, or why is that? So one hypothesis we had is just due to the really highly weathered nature of these soils actually allows soils to have some anion exchange capacity, which can hold onto anions, which is really different than what we see in a lot of the soils around here, where they actually typically have a negative charge to hold on to cations. And so even within the top 4 meters, and this is Kathy Jo Jankowski's work, which basically looked at the movement of nitrogen fertilizer in nearby agricultural systems. And she actually used the forest as a control system to compare the nitrate concentrations to. And they found that even within the top 4 meters, there's about 220 kilograms of just nitrate. And it's a big question as to if this is accessible to plants or not. But even if 1% of that was taken up by tap roots with water uptake, that would still contribute substantially to that increase in the leaf nitrogen pool. But under high weathering under high rates of weathering, you can get slightly positive charges on these soil particles that can hold on to nitrate. And that's one of the hypotheses that Alex Hiddell actually addressed in her dissertation. So, okay, so how common is this? Is this just unique to our site in the southeastern Amazon, or is this more prevalent across the tropics in general? And this is just a really rough mapping exercise, but here we have the broadleaf tropical forests mapped out in the green color, and then using the same types of soils, so oxisols and topographically flat, we find that maybe about 40% of the tropics are relatively similar with respect to soil type and topography. But that's not, a lot of these sites where we have these super flat, highly weathered oxisols are not the regions where we're measuring N fixation in general. So obviously there could be some bias in just where we take studies and the underlying ecoregions and variation in the types of soils that might contribute to some of the biases in our knowledge. So in summary, we found that fire does cause large nitrogen losses, but we didn't find that N fixation increases post-fire to replace those nitrogen losses. And I think it's going to be a big question as to if more intense disturbances are going to cause a tipping point, because we're seeing relatively high recovery rates in this forest and in other forests too, but it's unclear if that's going to continue into the future. So I just presented some of my dissertation work, and one thing that really surprised me was even working at Tanguru, which had really low phosphorus soils. I had heard about a fertilization experiment that Eric Davidson and others were involved with, where they didn't find super strong biomass responses, even when they added a lot of phosphorus fertilizer. So for my postdoc, I really wanted to work on how plants actually address nutrient limitation and to what extent plants can modify their environment. So just kind of moving away from a very traditional biogeochemistry perspective of the movement of elements and the availability of different elements rule the world, but rather some interaction between biology and then biogeochemistry. So for my postdoc, I really want to ask how plants address and overcome nutrient limitation. And if plants are able to address different nutrient acquisition strategies or just different nutrient acquisition strategies in response to changing nutrient availability and plant demand. And traditionally, when we think of nutrient limitation, we expect that higher nutrient soils should have higher productivity and higher biomass. And then we would expect that in low nutrient soils, we'd have lower biomass in soils. And this is a response from this is a meta-analysis of different fertilization experiments that have been conducted in the tropics. And zero effect size basically means no response. And what was really surprising was that in these old growth forests, summarized by Joe Wright, they basically didn't find relative growth rate tree responses in response to either nitrogen or phosphorus in mature forests, at least. And they did find a positive response in secondary forests. But this is really surprising, and it goes against what we learn in a lot of traditional ecology textbooks, that tropical forests should be really nutrient limited. And this also contrasts with a lot of this is a meta-analysis for northern deciduous temperate forests, and it's a little confusing because it's not a log response ratio, it's just a response ratio. But anything above 1 means that there's some response to nutrients. And in temperate forests, we actually see quite a bit of response to nitrogen or phosphorus additions, especially in combination. So why aren't we seeing these strong biomass responses to fertilizers in the tropics, but why do we see them in temperate systems? And one question or one one way to get at this is to think about if plants are able to allocate some of their carbon to acquiring nutrients or to use nutrients more efficiently, right? So it may be that in a low versus a high nutrient soil, you might have the same biomass, but you might have more investment below ground, or you might be using nutrients more efficiently. And maybe that's some way that plants can compensate for the soil nutrient availability. So plants can use nutrients more efficiently. They can invest in symbioses with mycorrhizal fungi. They can produce enzymes to break down organic matter, and they can also produce more fine roots, and nitrogen fixation is just one strategy I talked about. And we wanted to test if plants can address these strategies across changing demand, which the successional gradient is a really nice way to do that, because secondary forests have a really high nutrient demand compared to mature forests, right? So basically, plants are rapidly capitalizing after some disturbance event on the new light availability, and they're growing really, really rapidly. And eventually, their growth rates are going to slow down. And there's often a steady state, basically nutrient cycling of plants dropping their litter and then kind of using roots and the whatever's decomposed to basically take back up. So there's less mining of nutrients early on, or less mining of nutrients in mature forests relative to young forests. So you might expect that there's a really strong demand for external nutrients early on in succession. And in addition to the strength of nutrient limitation changing, the type of nutrient limitation can also change. And this is because phosphorus typically is less mobile than nitrogen. Nitrogen is really mobile. It's really easily lost after a disturbance event. And we know this from a lot of different experiments that have cut down forests and basically seen huge export of nitrate in streams. But then eventually nitrogen fixation is theoretically thought to kind of increase nitrogen availability. So you can basically get this shift across forest age or succession of nitrogen limitation to phosphorus limitation, in addition to this change in just overall nutrient limitation as well. And we wanted to investigate some of these different strategies, and two of the most important ones in the tropics, at least for phosphorus and or nitrogen acquisition, are root phosphatase and then investment in mycorrhizal fungi. So phosphatase enzymes are really critical in the tropics because most of the available phosphorus is actually going to be in organic forms. And basically plants and microbes can release these enzymes, which basically cleave phosphate groups off of organic matter that then plants can take up. But obviously there is some cost to producing these enzymes. On a mass basis, they're between 15 to 20% nitrogen. So you would expect that plants aren't going to produce phosphatase unless it's advantageous for them to do so. And the other strategy is mycorrhizal fungi, and they basically can explore more volume of soil, and they can also take up nutrients slightly more efficiently than some plant roots. And in exchange, they get carbon from the plant. So on average, maybe perhaps 20% of carbon or photosynthesized carbon is actually allocated towards mycorrhizal fungi. So you would also expect that plants are going to invest less in mycorrhizal fungi if they don't basically need them to access nutrients. And we wanted to test these strategies across a nitrogen by phosphorus factorial fertilization experiment in Panama that spanned 4 successional age classes. So from recut pasture plots that were regenerating and then that were theoretically 0 years old. They were about 4 years old by the time I went and sampled them, all the way to mature forests that are greater than 300 years old at Gigante in Panama. And you can really see just within 30 years, this change in biomass going from the pasture plots to the 30-year-old forest. The 30-year-old forests are really dense and they have very little light penetration coming through. And I'll talk specifically about one enzyme, phosphomonoesterase, and then to measure investment in mycorrhizal fungi, we used root staining, which is basically this idea that if you have more investment in mycorrhizal fungi, you're going to have higher colonization within your plant tissue or root tissue of mycorrhizal structures. So the more you're investing in mycorrhizae, the more mycorrhizal structures you should theoretically have in your roots. And our hypothesis was that phosphatase activity would increase slightly early on in succession as plants are mining new nutrients out of the soil and then decrease over time. And that maybe that nitrogen additions would stimulate phosphatase production because there is a nitrogen cost to phosphatase, and that perhaps phosphorus would only suppress phosphatase activity in the older forests because there's less or there's higher nutrient demand in those younger forests. So what did we find? Okay, so basically, I'm just going to show you the control treatments here in the white. And we did start to see an increase in phosphatase activity as we went from the 0 or 4-year-old forest to the 30-year-old forest, and a slight decline again in the mature forests. And we also found significant stimulation of phosphatase activity, like we hypothesized, with nitrogen additions in the 0 and the 10-year-old forest. So in the young forest, we did see that nitrogen increased phosphatase activity. But then we didn't see this effect in the older forests. So no nitrogen response in those older forests. And then we saw no response to phosphorus in the younger forest. So no suppression of phosphatase in the 0 and the 10-year-old forests. But we did see a significant response or a decline of phosphatase activity in response to phosphorus additions in the older forest. So there seems to be this interaction between the type and the strength of nutrient limitation across time that basically we hypothesized. So we basically saw all these responses that we predicted. And for mycorrhizal colonization, we expected that over time it would decrease or decline as plants theoretically are mining for less nutrients. And that maybe, mycorrhizae themselves can be nutrient limited. So maybe nutrient additions could stimulate the amount or the presence of those mycorrhizal structures in roots, and that response would decline across forest age. Well, what was really surprising was that actually mycorrhizal colonization across all the control forests stayed relatively high. And like so that was not what we hypothesized, but like we hypothesized, there was really no decline of mycorrhizal colonization in response to nutrients in the young forests. But in the older forests, we started to see this decline in response to nutrients. So there's also seemed to be some interaction of nutrient additions, but then also forest age, where we only saw a suppression of mycorrhizal colonization in those older plots. So in summary, we didn't see this decline like we expected, and no stimulation because the rates, at least that we measured, were really, really high, and some suppression, at least in the older forests. So in summary, we found that trees do adjust root phosphatase activity in response to changing nutrient and changing demand over time. And this seems to be relatively predictable and something that could potentially be incorporated into models. But at least for mycorrhizal colonization, we didn't see really predictable responses. And we also conducted a meta-analysis, and all of these results are really consistent with our meta-analysis. And so, I mean, part of it honestly could be due to some of the methods that we're using. So we also know that root staining is not the perfect way to actually measure carbon exchange with mycorrhizae. So if anyone has any ideas on how to better measure carbon allocation towards mycorrhizal fungi, I think that's something we definitely need to start working on and doing. But really, to actually be able to incorporate some of these, the flexibility of these strategies into models better, we have to really understand the cost of those strategies to the plant, and to what extent actually those flexible strategies are going to help alleviate nutrient limitation in the future under elevated CO2, as we predict that more nutrients are going to be bound up in biomass. So really big questions. Okay, so one thing that I'm really, really excited about now is thinking about the role of functional groups and how we can actually simplify species responses to different nutrient conditions and also community turnover. So I guess I just highlighted here, how basically we could expect that maybe if plants are investing more in nutrient strategies, we might not see any impacts on actual biomass or biomass limitation by nutrients. But the other thing that we haven't talked about is the role of community turnover. So you might expect that under high nutrient conditions versus low, you would have different species doing well and dying, and so such that you might have completely different tree species composition in a high nutrient site versus a low nutrient site. And we have lots of evidence that soils shape community composition across the tropics. Also from a nice gradient in Panama that I know Liza works at, across phosphorus gradient, they found that different tree species specialize in those different conditions. But predicting that is going to be a little bit more challenging. So, and what the consequences is that you might have the same biomass in a high nutrient soil versus a low nutrient soil, but just different tree species composition. And that is something that's going to be hard to predict. And the reason for this is because some of those different tree species might actually have different advantages under those different conditions, like those strategies that I had mentioned earlier. And I will preface this by, I'm going to move into temperate forests, and this was actually a pivot during the pandemic. So basically I actually worked in my backyard at the Carey Institute working on some of these questions, which was really nice. But the question here is asking if N-fixing trees can accelerate phosphorus cycling in temperate forests. And the reason for this is that temperate forests have always historically thought to be very, very nitrogen limited. And this might be changing across time because we're seeing some signs of declining nitrogen availability in some different forest sites in the Northeast. But at least historically, that there had been really high nitrogen deposition rates from fossil fuels and from agriculture in eastern forests, as indicated by this map here. So the warmer colors are indicating higher nitrogen deposition. And the reason that we're thinking that there might actually shift towards P limitation is because we're seeing a shift in just tree species composition in response to nitrogen deposition. So more arbuscular tree species relative to ectomycorrhizal tree species across FIA data plots or forest inventory analysis plots. We're also seeing declines in nitrogen to phosphorus or increases in nitrogen to phosphorus ratios in temperate forest leaves from Catherine Cowley's work. And then also from like the one nitrogen by phosphorus fertilization experiment in Bartlett experimental forest, we're also seeing growth responses to phosphorus and not to nitrogen for biomass. So some indication that there might be some changing nutrient dynamics or at least some co-limitation by nitrogen phosphorus at some of these sites. And you would expect that under high nitrogen enrichment, that nitrogen fixing trees would have less of a competitive advantage, right? Because now these systems are theoretically less nitrogen limited. And one of the most dominant nitrogen fixing tree species in eastern forests in general, there's low abundance of nitrogen fixing trees in eastern forests, is Robinia pseudacacia or black locust, early successional species here. And this is a nice map by Annika Stocconi that shows the distribution of some of these different N-fixing tree species across the continental US. So the purple here indicates Robinia. And what's really interesting about Robinia, and this also shows kind of the biogeochemical niche that some of these different plant species could have played under historical N limitation. But what's really interesting about Robinia is that from a survey of FIA data, it actually has one of the highest growth responses to N deposition and also high survival rates. So this goes against what theory would predict. So we would expect that theoretically under higher nitrogen deposition, there should be less of a competitive advantage for nitrogen fixing trees. Unless some of that carbon cost towards like nitrogen fixing bacteria is now going towards growth. That could be one explanation. But one question that we wanted to ask was if the dominant nitrogen fixer, Robinia pseudacacia, is doing well because it's accelerating phosphorus cycling. And the reason for this hypothesis is that historically nitrogen fixing trees have always been more phosphorus limited than other temperate tree species that are more nitrogen limited because they're able to allocate, or because they've always been able to alleviate their nitrogen limitation, that they might have some different strategies to get at phosphorus. And some of the dominant pools of phosphorus in soils are mineral forms, organic forms, like I mentioned, in the tropics, and then also phosphorus that is sorbed onto iron and aluminum oxides. And all of these different pools of phosphorus can then come into solution for plants to later take up. But there's also ways that plants can kind of modify their environment. And one way is that nitrogen fixers typically have a higher concentration of ammonium and nitrate in their soils, which actually can acidify soils. And so there's evidence that across a lot of nitrogen fixing plantations, that soil pH is lowered, and that can actually affect rock weathering rates as well. The other is that nitrogen fixers can be more mycorrhizal. There's some studies showing that they're very mycorrhizal. And the other is that they can allocate some of that nitrogen towards producing phosphatase. And there's some evidence or some theories that have predicted that the reason why nitrogen fixers are so abundant in the tropics is because they're actually able to allocate nitrogen towards producing phosphatase to get at phosphorus. And we wanted to actually compare some of these strategies or functional groups across or in response to nitrogen and phosphorus in both an using an experimental approach and then also an observational approach. So experimental being a greenhouse study. So we wanted to compare the phosphorus acquisition among some of these dominant arbuscular and ectomycorrhizal tree species across temperate regions. And we used like black locust or Robinia, sugar maple, oaks, cherry trees, birch trees, etc. And then we fertilized across 3 levels of nitrogen to represent a gradient in nitrogen deposition. And then we grew the plants in a nutrient-free medium. It was a turfus that we added 2 sources of phosphorus to. So we added litter bags as a source of organic phosphorus, and then apatite rock as a source of inorganic or mineral phosphorus. And then we replicated this experiment and then grew the plants for about 18 weeks. So you can really see how much the plants grew across 18 weeks. And then we wanted to compare some of these different strategies here. And I don't know if anyone knows Brandon Radcliffe here. He's a Master's of Forestry student, but he actually worked with us at the Carey Institute, helping us harvest this experiment. So I won't talk about some of the methods here because they're the same that I used in the last, or talked about in the last portion of the talk. But we did look at total phosphorus uptake in the tissues, and we did find that Robinia pseudacacia took up the highest amounts of phosphorus. So this is what we hypothesized, that maybe they'd be better at getting at phosphorus. And at least in terms of tissue accumulation over time, we did see higher rates of that. And then because we had those rock bags that were buried in the pots, we were able to look at a mass loss change, and we didn't see any differences, at least in the time scale that we were looking at, of any mass loss change of that apatite material. So no differences in rock weathering across the different plant species and across different levels of nitrogen. And then we also looked at mycorrhizal colonization across some of these plants, but we didn't necessarily find that Robinia was the most mycorrhizal, and that wasn't the reason why it was able to take up most of the phosphorus. But we did find that on a per gram of root basis, Robinia had orders of magnitude higher root phosphatase activity than all of these other tree species. And then when we scaled it to the actual fine root biomass of all those tree species, way higher phosphatase rates under Robinia species or this nitrogen fixing species. So we know that greenhouse experiments can be artificial, and they are focused on young plants. So we also wanted to compare some of these different phosphorus uptake strategies or acquisition strategies across mature trees in the field. And so we were able to find 3 forest sites that had at least 2 individuals of all those 6 of the 7 species that we looked at. And then we traced their roots and then measured soil characteristics. And we used those roots to measure those strategies. And I don't know if anyone's done any root tracing here before. A few nods. Okay, well, oops. This is one example of a video of a pine that we traced roots from. So this actually took over a day. So if anyone wants to come and trace roots sometime, I'm happy to get volunteers. But this is just one species, and this was just like one root that we were actually able to get from the base of the tree. And really the challenge here, if you want to measure anything and identify it to an individual species without using barcoding, it's really hard to actually isolate a specific root to a specific species, right? Okay, so, and then because we had those apatite bags, we also buried them in the field for over a year. So we were able to look at mass loss changes. And what we found was that there wasn't any differences in mass loss across the species, except for sugar maple, which I think it's because of the calcium in apatite, and sugar maple demands a lot of calcium. So that could have been one of the reasons why there was higher weathering rates or mass loss rates for sugar maple, but basically no differences, at least in mass loss. And then and this could be due to the fact that we didn't see any differences in soil pH. And because we were working in really mixed forests that were not monodominant, whereas I think a lot of the historical studies that had found differences in soil pH were done in more monodominant sites. But there did seem to be some relationship between soil pH and weathering that I think further needs to be investigated. But we did find that at least most of the arbuscular mycorrhizal tree species had really high colonization rates in general. So it does seems to be way more mycorrhizal in the field than at least in the greenhouse. And because black locust is like an early successional species and it tends to do well on edges, I will say a lot of the Robinia species that we sampled, because we wanted all the trees to be co-located in the same sites, they weren't the happiest trees. And so we didn't actually find differences in root phosphatase activity in some of those older trees that were Robinia. So this was different than our seedling experiment. But then we did find, then we actually went and sampled some of the saplings, and we did find slightly higher root phosphatase activity in the saplings. So in conclusion, we didn't really see differences between Robinia pseudacacia and all of these other tree species in terms of the soil pH or rock weathering or mycorrhizae, but there does seem to be a difference, at least in those younger trees, for phosphatase activity. And that could explain why they were able to uptake more phosphorus, at least in our greenhouse experiment. And I think it still remains to be a question if they're actually doing well under high nitrogen deposition because of phosphorus or because of some other reason. But basically, this is an approach to kind of get at some of the actual function behind some of the mechanisms that we're seeing in tree species turnover. So in summary, Robinia did seem to accelerate organic phosphorus cycling through phosphatase, but again, it really remains to be seen if N fixers as a group are functionally similar. And I think if we want to make any claims on phosphorus cycling, we'd have to really look at phosphorus flux as a whole and not just nutrient uptake in specific roots. So yeah, it was really exciting to find that plants are able to adjust strategies on some relatively short time scale at the field level and across community groups. But I think these are really interesting questions that I'm excited to pursue further. And I don't think anyone's necessarily going to be able to answer in their lifetime if the land sink is going to continue to take up carbon at the same rates indefinitely, but there's a few steps that we can probably take to get there. And the first is, at least for the nutrient limitation aspect, nutrient concentrations alone don't indicate limitation. I think we're going to have to do a lot more work with actual plant uptake and stoichiometry of plant tissues to get at nutrient limitation. At least at this moment in time, we seem to find relatively rapid rates of recovery across ecosystems to disturbance, but there is a lot of spatial heterogeneity in the tropics that I think we're just starting to work on getting a handle of. And the other conclusion is that plants can upregulate nutrient strategies. But in the future, I'm really excited to think about what are the spatial drivers of spatial heterogeneity, both abiotic and biotic, and then also asking if ecosystems are going to continue to recover rapidly, how plant communities can respond to nutrient limitation, and how nutrient limitation interacts with other drivers such as drought, and coupling above and below ground responses. And with that, I'd like to thank my PhD advisor and postdoc advisor, Bob Howarth and Sarah Batterman, and then a lot of the other folks who helped with the research as well as our funding sources. And I think it's probably the end of the class period now, but I'm happy to take any questions if there's time.
Great talk, and it's really cool work. And I'm amazed by that picture of you in a meter down with bats around and everything. Seems like not easy work to do. I'm just wondering, as we think about shifting from sort of like a law of the minimum to multiple element limitation sort of framework, like how do you know when you've looked at enough potential limiting resources? Like, how do we know that it's molybdenum and not vanadium that's limiting nitrogen fixation or light availability or what else? Like, how do you know when you've looked at enough things?
Yeah, this is a really challenging question. I think like one of our challenges is that we rely on fertilization experiments to ask those questions. And there's also different aspects of limitation. Like you can have a growth response, like at the Melanahe plots in Bartlett to phosphorus, but then their roots are responding to nitrogen, for example. So what does nutrient limitation really mean? And I don't know if I have really an answer for you, but I do think one of the big questions is actually trying to understand, or one of the big ways moving forward is we're going to have to really look at like tissue stoichiometry and uptake, because even like looking at, I guess one of the challenges is that whatever we look at for extractable nutrients in the soil doesn't necessarily reflect potential uptake towards organisms. And there's a lot of challenges with fertilization studies, which is going to be like the proximate versus long-term ultimate nutrient limitation. And yeah, so I don't have an answer, but I do think we don't even really know wood uptake. So wood stoichiometry is a huge opportunity, for example. Thank you. Liza?
Sorry, I'm wondering how N-fixer abundance is compare in the southeastern Amazon compared to other areas of the neotropics. And if you map that kind of distribution of molybdenum, hard element to pronounce, molybdenum concentrations and arrival through dust and fixer relative abundances across those areas, if there's any correlation.
Yeah, that's a great question. I think in most mature neotropical sites, the N-fixer abundances are low. And I think even like at our sites in Tanguru, we got to about 10%. I think in Panama and some other sites in Central America, the highest abundance at some of the early successional sites is about 15%. And yeah, a question of like why if they're there because of higher molybdenum. So the interesting thing is that in Asian tropical forests, you actually have a really low abundance of N fixers, but relatively higher rock-derived nutrient availability. There's really high erosion rates across a lot of the tropics. And I think one of the hypotheses as to why there is lower nitrogen fixer abundances in the Asian tropics is because of potentially rock nitrogen in the sedimentary rock that in the Asian forest. So I think that there's a lot of interesting feedbacks that would be interesting to investigate. Thank you.
All right, thanks, everyone.